So good afternoon to you all, and welcome to the intelligent automation of high-throughput chromatography for enabling wide adoption of HTPD workflows in vaccine process development. My name is Salome. I'm one of the Vax-Hub researchers, and I will be your host. Before we start, just some housekeeping rules. As mentioned on the invitation, this webinar is being recorded. If you have any concerns, please let us know and we will make you anonymous. Also, during this session, and to avoid any connectivity issues, please keep your microphone and camera off. This webinar will allow us to understand the efficiency of high-throughput technologies on the development of live purification steps using chromatography. And at the end of the talk, there will be a Q&A session. And to participate, either raise your hand or type your question on the webinar chat. Due to time constraints, if your question is not answered or if you have a late question, feedback, comment, please feel free to send myself or Anne an email with all of that. Without further ado, it is my pleasure to introduce you to our speaker, Dr. Spiridon Konstantinidis, who is a senior scientist at MSD's Vaccine Process Development Department. Spiros, up to you now.
Thank you very much, Salome, and hello, everyone. So, Salome, again, thank you for the introduction and also for giving us this opportunity to share with you some of the work we have been doing in terms of automating some high-throughput chromatography technologies that, again, has enabled us to adopt widely a high-throughput process development approach in the area of vaccines process development. So before I start with the bulk of this presentation, I would just like first to introduce to you our company. So we are known as apologies, let me just share very quickly the screen. Okay, now you should be able to see it.
Yes.
Excellent. So we are known in the United States and Canada as Merck, whereas in the rest of the world known as MSD. And our company is one of the few remaining companies that is actually dedicated to the business of researching and producing vaccines. And as a matter of fact, we are actually making investments in our pipeline and also our manufacturing capabilities to help protect an additional 1 billion lives by the year of 2030. Our company is also highly committed to innovation. And as a matter of fact, we have more than 100 years of history in terms of developing and delivering vaccines. And perhaps the best personification of our commitment to innovation is in the face of our former leader, Dr. Maurice Hilleman, who throughout his career, he helped to develop more than 40 vaccines for use in both humans and animals. And as a matter of fact, our former MSD leader has been credited with saving more lives than any other medical scientist in the 20th century. So continuing with this theme of introductions, I will spend the next few slides to tell you a few things about our organization, which is MSD Vaccines Process Research and Development. And then I will touch base briefly on the different challenges we typically made within the area of vaccine process development. This will lead me to introduce to you our high-throughput process development team and the different workflows and tools we have developed in order to meet the aforementioned challenges. We'll then demonstrate the deployment of these tools through some brief case studies, and then we're going to wrap things up with a summary and conclusions. So our organization falls under the umbrella of MRL alongside with other organizations, including biologics, small molecules, process research and development, and analytical research and development. And within our organization, we have 3 departments. We have the Department for Vaccine Drug Product Development, the Department for Bioprocess Clinical Manufacturing and Technology, and finally, the Vaccine Process Development Department. And all of these 3 departments are located in one campus, which is at West Point, approximately 30 miles from Center City, Philadelphia. The campus is also MSD's largest pharmaceutical manufacturing facility. So the work we are doing in our department, which is vaccine process development again, is actually spanning the different stages of bioprocess development. So we're doing work from late discovery and early preclinical studies all the way to phase 3 clinical trials and early manufacturing. And the responsibilities we have within our department span different aspects of things such as the design of processes, the development, the characterization, the generation of drug substance material that can be used for to assist the development of analytical methods, and ultimately the scale-up and tech transfer of complex bioprocesses. And our overarching goal is to deliver transformative and life-saving vaccines using the best science. So within the area of vaccine process development, we're typically dealing with a wide range of vaccine biomolecules, and these biomolecules can also have varying degrees of complexity. So on one end of the spectrum, we have the polysaccharide vaccines, which are relatively simple, and on the other end of the spectrum, we have live virus vaccines, which are rather complex. And a lot of the things I'm going to be discussing with you today is going to be focusing on our work with live virus vaccines. Now, because the vaccine modalities are rather diverse and complex, it means that most of vaccines, there are of course few options such as DNA vaccines. And this means that every time we have a new product coming in our pipeline, we have to define new processes in a de novo fashion. And this is what creates a lot of the challenges we meet during vaccine process development. So for example, due to the lack of platform processes, when we're doing upstream process development, we need to spend a lot of time in screening different types of cell lines. Because due to virus tropism, we don't have one particular cell line that will actually guarantee the good production of all of our viruses. And of course, with screening different cell lines, we have going to be having different levels of product titer. We're going to have different levels of impurities, including host cell proteins, and also different identity of impurities of, again, such as host cell proteins. And this, of course, creates another level of complexity that it's not typically met with other biopharmaceuticals that are actually benefited by the existence of platform processes. Now, when we're doing work with developing downstream purification processes, we're typically working with either digestion flow, filtration-based unit operations, or chromatography-based unit operations. From these two, TFF is perhaps the easier one to try and develop, but there's always the question of whether TFF by itself can actually deliver a final product satisfying our purity specifications. And in case it does not, then we have to actually try and develop a purification process using alternative technologies such as chromatography. So when we're doing investigations for a chromatography-based purification process, we're actually testing all sorts of different types of chromatography media, including membranes, beads, and monoliths. And for each one of them, we are testing different chemistries of ligands, and we're also evaluating them in different modes, including both flow-through and bind and dilute. Every time we're doing these type of investigations for a chromatography-based purification process, we always keep our mind on a couple of questions. So for example, even if we come up with some very nice capture condition steps, a condition for a capture step, there's always the underlying question of whether we're still going to be able to get a good elution yield from this step. In case we do not, of course, we wouldn't be able to use it for a manufacturing process. And at the same time, we also ask ourselves whether the conditions, the mobile phase conditions we're going to arrive through our investigations, whether they're going to have a negative impact on our product or on our live virus through, for example, inducing aggregation or having a negative impact on its effectivity. Now, alongside with this type of investigations, we're also trying to evaluate alternative technologies for a purification process, focusing on, for example, solidification or precipitation. But typically, this type of technologies do well within a lab scale environment, but they don't translate that well at manufacturing scale. Now, adding to these type of challenges, we also have analytical challenges, and especially in the case of live virus vaccines, our main product tracking assays are cell-based assays, and this is what actually makes them to be quite complex, slow, and at the same time, they typically have large amount of analytical noise already embedded into them. And this, of course, creates a lot of difficulties when we're running our experiments. So all of these challenges together, they essentially create a situation wherein we often have to navigate through some rather large experimental spaces. And these spaces are truly multi-parameter, complex, and dense. And this means that we have to screen also a very large number of operating conditions. Now, because our business is undergoing a nice growth over the last years, our project timelines are quite aggressive, and that also means that we have to deliver the results from such screens fast. So all of this together, they create a rather challenging situation, but at the same time, they present a prime opportunity to both leverage and showcase the application of high throughput process development to the development of processes for vaccines. And the reason behind that is, of course, the fact that HTPD was actually conceived as a way to actually address these kind of challenges and work with these kind of challenging situations. So in our department and our high throughput process development team, we're trying to meet these type of challenges where we're doing work in both upstream and downstream process development. Now our team is a relatively small team, so we have 5 permanent staff members and 4 contractors. And the work we are doing is actually spread across 2 laboratories. And within these labs, we have a lot of different pieces of analytical equipment, because I should point out that we are actually responsible for developing, deploying, and supporting our own analytical methods, meaning that we rarely actually submit samples for analysis to our colleagues over at the analytical department. And of course, since we are a high throughput group, we also have a lot of robots in our labs. Now, in this case, when we are talking about in the science fiction sense, but instead we are talking about liquid handlers. So we have a good selection of robotic systems from different vendors, including Ticker and Hamilton, and we also have some large integrated robotic systems from high-res biosolutions. And of course, our upstream colleagues, they have access to Amber stations and also a compact T robotic station for work with flash-based cell culture development. So the overall goal of the high-throughput process development team is to actually use our state-of-the-art robotic systems and our in-house developed analytics within our workflows. And we're doing this in order to leverage our high-throughput technologies and enable best process at first filing. Now, within the area of downstream high-throughput process development, our HTPD workflows have 4 main cornerstones. So we're actually trying to fabricate our own custom robot hardware so we can actually implement automated workflows in the way we envision them. And we also have generic robotic processes that also use fixed configurations, meaning that we're trying to avoid any situation wherein from one experiment to the next, we have to completely change the way that the robotic station looks like. Our robotic stations are also flexible because they actually allow for on-the-fly robot programming. And we have also developed our own little piece of software for programming robots and also for doing data analysis. So what we are trying to do within our team for downstream process development is to use our workflows in order to implement walk-to and walk-away automation that is also accessible to pretty much everyone within our team, even to automation novices. And by doing this, we're hoping to reduce robotic stations and high throughput experiments to routinely deploy tools. And this will allow us to essentially focus on the interpretation of experimental results instead of focusing on the act of running experiment itself. And ultimately, we are going to unlock the potential of high throughput process development and its benefits to the process development for vaccines. Now, within the work we're doing for developing chromatography-based purification process for vaccines, we're using 2 main high throughput chromatography techniques, the 96 well plates and also the miniature columns. So between these 2 types of techniques, we're generally speaking generating towards the RoboColumns. And the reason behind that is that they are actually quite easy to scale up. And at the same time, it is very convenient for us that we can have them packed with any stationary phase we desire upon request at Repligen, for example. Whereas in the case of the plates, we will have to prepare our own slurry plates if we want to test many different resins from different manufacturers in one experiment. So we're doing this, we're deploying these high throughput chromatography technologies, and we're supporting our process development experiments using 2 robotic systems. The first one is a Tican Evo 200 that is fitted with disposable tips, and this is essentially a Swiss Army liquid handler. So we're using this robotic system to do a lot of different activities. So for example, we're using it to deploy our mix and read assays, which we also term as on-deck analytics. And we're also using this robotic station to do some sample handling and preparation of samples for deployment of analytical methods, such as quantitative Western blotting. And of course, as you can see in this photo, we're using this robotic system to run our high throughput batch chromatography experiments, in which case we're using slurry plates, membrane plates, monolith plates. I would like to point out that, as you can see from this photo, this is a standalone robotic system, meaning that we don't have any, let's say, sophisticated or expensive solutions for plate storage. Despite this, we are getting a very nice and decent throughput from this robotic system, as it looks in this photo. The downside of that is that, of course, that as you can see in this photo, we're dealing with some rather busy deck configurations. As a matter of fact, in this photo, we're pretty much using every grid on the robot's deck that we could use, that it is indeed available to us. The second robotic system we're using is a Tican Evo 150, and this is fitted with fixed tips. And again, as you can see from this photo, it is primarily used to actually run RoboColumn-based experiments and to also support them by the preparation of buffers and solutions. Since we have a plate reader integrated to this robotic system, we're also using this to also support some plate reader-based measurements. And again, as you can see from this photo, we're dealing with a standalone robotic system that it has, again, a rather busy deck. Despite this, and despite the fact that it's actually a small system, we can again get a rather nice and decent throughput out of it, even when running these RoboColumn experiments by using deep well plates for collection plates instead of using the traditional microplates. In this slide, I'm sharing with you a flowchart of how we typically deploy and run our high throughput chromatography-based experiments for process development. So this slide also contains our different workflows that actually make the entirety of running high throughput chromatography investigations within our labs. And this workflow has essentially four main parts. So we typically start our work with coming up with a study goal, and this could be the goal of one particular experiment or of a campaign of experiments. Once we have this, we then use an experimental design approach to start defining our inputs and our outputs. And based on this, we start preparing our buffers and solutions that are used to run our experiments. So we use these buffers and solutions with our plate chromatography or RoboColumn chromatography experiments. And during these experiments, we start generating fraction plates. And typically, we measure these fraction plates as they're being generated part of the experiment. At the end of the experiments, we can then further process the collected fraction plates by aliquoting them or diluting them or even pooling them. And this way we generate a second batch of plates. And then we can analyze both batches of plates with our analytics before we start processing our results, the generated results. So by the end, when we process all of our results, we can then start understanding whether we have met our study goals or not. And depending on the answer, we can plan the next round of experiments. So I will spend the next few slides to talk you through the different bits and pieces of the different workflows we deploy when we're running these high throughput chromatography experiments. And the first part will be to talk you through how we're actually supporting some advanced sample handling for our high throughput chromatography experiments. So here we're actually doing some complex liquid handling experiments by using our own custom VBA tool. So if you're wondering why we need to have a custom tool to just perform some liquid handling experiments, well, the answer to that is that Tikan's Evover, which is the software that Tikan is using to operate the robotic stations, is a rather powerful piece of software when we want to carry out some simple and relatively repetitive actions. However, when we want to have some more challenging liquid handling experiments and we want to carry them out in a generic fashion, it becomes rather cumbersome to use. So we have tried to meet these challenges by coming up with our own little piece of software. And we're using this to provide both assay support and also screening support. So this software has a lot of different interesting features. And here I have just highlighted some of the main ones. So for example, it is compatible with all of the Evo robots we're using within our labs. So it's not only tailored to one particular robotic system. Instead, we can use it for all of the Evos that we have that we're doing work with. It can control all the arms we have in our robotic systems, including the liquid handling arm, the robot manipulator arm, and the multi-channel arm. It can also, for example, do some automatic aliquoting of large volumes, which is rather useful to us when we're actually generating buffer stocks in use for our experiments. And it also provides with some creature comforts. So, for example, it can detect errors that would otherwise go unnoticed through Evover. It is also integrated with Evover's databases, meaning that it will give us drop-down menus pre-populated with a selection of liquid classes, carriers, and libraries that are available to us. And because, as I've already said, a lot of the robotic systems we're using, they have rather busy deconfigurations, we have implemented in this software some sophisticated procedures to actually help us maximize the deck utilization. The way that this tool works is that we can start it on our personal machines and we can start defining our inputs, detailing how we're going to run our liquid handling experiment. And while we're doing this, we're making use of the different selection of advanced features included within this tool. And once we are happy with our selections and our inputs, we can launch it. And once we do this, we essentially get a list of files generated within a predefined location. So in these files, we have a spreadsheet, which is essentially a log of how we set up our experiment. And we also have this .tsc file that I'm showing to you on the right-hand side is essentially an Evover script, meaning that we can actually generate through this tool Evover scripts that can be loaded and executed by robotic systems from scratch. And of course, we have all of these GWL files. These are work lists that are generated again by our tool, and they contain all the different commands that we need in order to complete our experiment. And you can see this example down here that essentially we're having aspiration dispensers and washers. And that is just an example of one of these work lists. Once we have all of these files generated, we can then go to our robot, essentially load up the newly created Evo script, put our plates in the robot, and launch it. And by doing this, we're essentially having a situation wherein we can spend maybe up to 30 minutes of tool time, followed up with 10 minutes or 5 minutes of robot time to start carrying out experiments that could have a duration of up to 6 hours. And we're doing this without requiring any robot programming at all, which means that we can have essentially some rather complex experiments being carried out even by people who have never used robotic systems before. The only thing that they need to know how to do is to use an Excel spreadsheet, basically. So in the next few slides, I will share with you one of the applications that we're actually using this little software of ours, and that's actually being used quite a lot within our experiments. And this has to do with the pulling of chromatograms. So what I have here for you is a cartoon example of chromatographic separation. And you can see, for example, that we have the load followed up with a wash, and then we have the elution gradient, in which case we have the elution of 2 peaks, and then that's followed by a strip. And in this case, for example, there is one more peak coming out towards the very end of the strip, which could perhaps be a strongly bound impurity. So typically, when we're actually running this type of experiments, we can actually collect fractions across the different stages of this chromatographic separation and place them within a 96-well plate, which we are going to term as a fraction plate. So if we wanted to understand what happened during this chromatographic separation, we could have, in this case, to analyze up to 81 samples. So instead of doing this, what we do quite frequently within our lab is to actually try and break this chromatogram into smaller pools. So what we do is we can actually aliquot the fractions that were collected during the loading of that plate into one well of a separate plate, which we're going to term pool plate, and we can do the same thing with the fractions collected during the washing phase of our column. Then, for example, during the elution, we could aliquot the small volumes from the fractions collected during the elution of the first peak in one well of our pool plate, and we can do the same for the fractions collected during the elution of the second peak. And of course, we can follow the same procedure with the fractions collected during the strip. This means that by doing that, we can actually go from having to analyze 81 samples to having to analyze up to 6 samples and getting a very good idea regarding how our experiment worked. So this helps us quite a lot with managing the sample load that we have in our experiments. So what I have in this slide over here is just an example showing to you how this tool looks like in this particular example, in this particular application. So we have down here the plate maps for the fraction plate and for the pool plate, and you can see that essentially they match the little figures that I have up here for this application. And on the left-hand side over here, we have all the different pieces of information required to actually program the liquid handling from the fraction plate into the pool plate. Now, this is a relatively simple example, but you can imagine that when we're running, for example, RoboColumn experiments, in which case we could have up to 8 RoboColumns at a time, we could have 8 chromatograms looking entirely different to one another. And in this case, being able to actually generate pools from 8 different chromatographic separations can become rather challenging. Yet, this is something we can do quite easily with our developed tool, and it can be done even by people, again, who have never worked with robotic systems before. The only thing, again, that they need to be able to do is to use your tool or use essentially an Excel spreadsheet. So with this, we've gone through one of the workflows that we typically use within our high throughput chromatography experiments. And the next thing I would like to discuss with you is how we deploy our on-deck analytics. So in this case, we're talking about high throughput analytics or essentially mixed rate assays. And we do this by having an advanced Evo script. So what we have come up with is an Evo code that's approximately 2,700 lines in length, so it is a rather long script, and it has a lot of interesting advanced features. And some of the most useful for us is that it already has predefined method settings for the most popular assays we deploy routinely in our laboratories. And this could include, for example, nucleic acid assays or total protein assays, and etc. At the same time, with this tool, we can also generate calibration curves on the fly, and this can be done by only one vial, one standard. And these calibration curves, of course, they're going to be multi-point and also done at various levels of verification. And finally, we can also dilute our plates that we want to analyze with having different dilution factors across the different plates. So even though this whole process appears to be simple, it's still very, very useful to us because by the end of the day, we can actually generate our different plates and then we can load them up onto a robotic system and deploy this tool and start analyzing our plates overnight. And then the next day in the morning, we can already have results. Based on the results, we can start seeing how our experiments worked, if they worked or not, and etc. And even start planning any, let's say, future activities based on these preliminary results. And at the same time, we are also supporting different, let's say, analysis of samples submitted to us by our sister groups within our department. So this is not only useful for us as a high-throughput process development team, but it's also useful for the entire department. So with this, now we can have a look at how we actually run plate chromatography experiments within our laboratories. So in this case, we're trying to do flexible and accessible plate chromatography screens by using our in-house developed VBA tool and also again with some advanced eWork programming. So again, if you're wondering why we need to have such a custom tool to run just plate chromatography experiments, well, the answer to this is that amongst the different high-throughput chromatography techniques, plate chromatography is perhaps the most challenging to automate. And as a matter of fact, this automation challenge becomes even more challenging or even more increasingly more difficult when we consider that we have different types of chromatography plates from the different vendors that need to be run in different ways. And as a matter of fact, even the fact that they have small difference in terms of the dimensions are things that they need to be taken into account when we're actually running them in fully automated workflows. At the same time, with this type of high throughput chromatography technique, we can have a scenario we can run many different studies. So we've tried to actually meet these challenges by coming up with our own, again, in-house developed tool, which has, again, a very nice selection of advanced features. So for example, it allows us to implement walk-away automation for pretty much every type of study we could perform with this high throughput chromatography technology, including the screening of binding and wash conditions and elution conditions, the measurement of isotherms that could use multi-load experiments, and even the measurement of absorption and desorption kinetics. At the same time, this tool is compatible with a good selection of chromatography plates from the different vendors. And as a matter of fact, it already contains predefined settings in terms of how we can actually run these plates. And it can also control all of the arms on our robotic systems and all the different devices that we use when we're running these plates. And this provides us with a lot of flexibility because, for example, within one experiment, we could have one part of the experiment wherein we're handling our chromatography plate using a multi-channel arm and we're emptying it by using our vacuum manifold. And then for another part of the same experiment, we could be liquid handling our chromatography plate using our liquid handling 8-channel arm while emptying them by using our centrifuges. Now, another thing that is very challenging with this high throughput chromatography technique is the fact that if we want to be able to run it in a robust fashion, we need to have some on-the-fly calculations. We need to be able to perform some on-the-fly calculations. So what we are talking about here when we're discussing about the robustness of this high throughput chromatography technique, it actually pertains to the fact that we want to make sure that we're actually collecting all of the fractional volumes we're actually loading into these plates. Because what can happen is that you can actually start this experiment with these 96 well chromatography plates and then come back to see how things are going to only find out that part of this plate, some of the wells of this plate, we were not able to actually empty them completely as we're going through the different stages of the experiment. And this means that we can start having a lot of splashes happening and even pools of liquids on the deck of our robot. Now, in our case, because we're doing work with live virus vaccines, it means that we are working with biosafety level 2 material, and this is something that we cannot have happening within our labs. We do not want to see any pools on the deck of our robot that contain viruses. So what we have done within our processes, we have implemented a little logical loop. So every time we generate a fraction plate, we immediately measure it using our plate reader, and then we have a little software that actually will get in there, process the different plate reader measurements, and calculate fraction volumes. And it will then compare the fraction volumes against the limit. And depending on the results of this comparison, If things have not worked well, we're going to get a prompt for a user input. Otherwise, it will continue to actually generate the remaining fraction plates in our experiment. And by doing this, we can ensure that we're running these experiments robustly by eliminating any incomplete collection of fractions. And again, because we're working with BSL-2 material, this is very important to us. So the way we're actually using this tool in our laboratories, we launched it again on our personal computers, and we started setting the different inputs and detailing how we're going to run our experiment. And again, we're making use of its different advanced features. Once we are happy with our settings, we launch it. The tool itself will generate a list of files. And again, here we have worklist files that contain all the different commands that are needed by the robotic system to run the experiment. And as you can see here in this example, we have primarily advanced worklist commands, in which case we're controlling the multi-channel arm, we're controlling the robot manipulator arm, the shaker, and also the vacuum manifold. So once we have these files generated, then we can go to our robot and load an evolver script that is particular to the type of chromatography plate we're running. We put our plates inside the robot, and then we run it. And by doing this, we have again a situation wherein we can spend approximately 10 minutes of tool time and no more than 10 minutes of actually putting things and setting up things on the robot and carry out some rather complex experiments that could have a duration of more than 8 hours. And again, we're doing this without requiring any robot programming from the end user. And this means that we can have, again, any automation novice who can actually walk to our robotic systems, set up their experiment within minutes, and then start running it. So we're really making accessible all of these type of experiments to pretty much any member of our team, regardless of their experience with working with high throughput experiments and robotic systems. So with this, we have gone through most of the parts of the different workflows we're using when we're carrying out high throughput chromatography experiments using 96 well plates. So I think it's going to be quite useful to show to you how a typical example looks like within our labs. So typically by designing a different chromatography studies, we can end up with having to prepare up to 5 plates. So these plates typically contain our starting material in a load plate, which is again prepared at different, for example, mobile phase conditions. And then we can have a plate with different equilibration buffers, another plate with different wash buffers, another plate with elution buffers, and a final plate with different solutions used for stripping, for example, the monolith plates or the membrane plates or the slurry plates. And we use these plates to run typically one chromatography plate. And part of this experiment, we're going to generate up to 11 fraction plates. And as these fraction plates are being generated, again, we're measuring them on our plate reader. So typically from this kind of experiment, the last 8 fraction plates truly contain fractions. Typically the first 3 only contain our equilibration buffers. And what we then do is to take them and process them further by aliquoting them or diluting them. And this means that by the time we start deploying analytical methods, we could have up to 40 plates to analyze. And by analyzing all of them, we are ending up with around 4,000 data points that we need to process before we can even begin to understand what happened in our experiment, how our experiments worked. So we quickly recognized that essentially we had to provide the backend support for all of our high throughput chromatography screens so we can actually facilitate this stage of data processing and review. And this is something we've done in our labs by coming up again with our own little piece of software that help us to extract data from the most popular assays we use in our labs. So what we can do with this tool is that as soon as we have completed our assays, we can use it to get all the data out and have them tabulated in another easily readable manner. And by doing this, we can start investigating trends, understanding how things work within our RAM, and then we can start planning the deployment of lower throughput analytics to try and analyze all of our samples. And by doing this, we can actually achieve a timely completion of studies. So we have arrived essentially by using this workflow in a steady state wherein we can actually have approximately 1 week of turnaround times for such experiments, even when these are being supported by only 2 scientists. So with this, we can now proceed to having a look at how we actually use these RoboColumn chromatography experiments within our labs. And we have 2 main parts, the preparation of elution buffers and then the actual RoboColumn chromatography experiments themselves. So first, we're going to focus on the preparation of elution buffers. And this is important because when we're running RoboColumn experiments, we don't have a traditional, let's say, benchtop chromatography station that can actually generate gradients in on-the-fly fashion using a gradient mixer, in which case you can mix, for example, buffer A and buffer B. Instead, we need to have all of the buffers ready to go before we start running an experiment with RoboColumns. So to do this, we have again developed our own little standalone tool that's based on VBA. And again, it can use different user inputs to create elution methods for up to 8 RoboColumns at a time. In this case, our elution methods can be anywhere from isocratic, step, multi-step, or even gradient elution methods. And this is very useful for us because by using this tool, we can actually genuinely test up to 8 entirely different chromatographic separations in parallel within one RoboColumn run. And what I have here on the right-hand side is just an example of how, for example, different gradient elution methods could look like by using our tool. And you can see we have 4 columns in this case, and we have one sort of gradient that has a relatively steep slope, a medium slope, and a shallow slope. And for one of these gradients, we also have a case where we're going only up to 50% B, as opposed to going all the way to 100% B for the rest of the columns. So again, this tool has been very, very useful to us because, again, we don't have any limitations regarding how we can run these RoboColumn-based experiments. We can, again, pretty much have any type of chromatographic separation we desire within an 8-column RoboColumn run. So regarding how we're actually running our RoboColumn experiments themselves, again, we're trying to do screens in a flexible and also accessible fashion. And to do this, we have developed, again, our own little standalone software, and we're also deploying some advanced EVOR programming. So if you're wondering, again, why we need to have our standalone software for RoboColumn experiments, well, the reason behind it is that the software provided by Tegan themselves, which is called HT Chromatography Wizard, and both a completely manual approach of hard coding methods to run RoboColumns, they have, of course, their own strengths and weaknesses. But what we have done in our approach is to essentially combine the strengths of either of these two approaches while eliminating the weaknesses. And this is something we have detailed in the literature with a publication back in 2018. So what we do when we're running these RoboColumn-based experiments is that we can open a RoboColumn script on our robots, we can put the columns in the different plates, or we can start running it. The script itself will then ask us for the insertion of some end user inputs, and these inputs will essentially describe of how to run up to 8 chromatographic separations at a time. Then we're using a software to essentially convert all of these inputs into all the instructions that are required by the robotic station to run our chromatography experiment. And then the same software will also be used to generate a report at the end of our experiment. And here, what we mean by saying that we're generating a report, we're essentially generating this results text file, which contains both raw measurements and also process measurements from the different fractions we have generated during our chromatography experiment. And we're also generating chromatograms. We're plotting chromatograms. And this means that essentially within a few minutes from the completion of our experiment, we can start immediately observing trends and even finding out if our experiment worked and how it worked. And this is, again, very important to us because by doing this, we can immediately and very fast start planning any follow-up activities based on the results we got from such a RoboColumn experiment. So as we've done in the case of the 96 well plate chromatography process, I think it's going to be quite interesting for you to also show you how a typical RoboColumn experiment looks like in our labs. And we are finding ourselves facing a situation more and more frequently wherein our experiments can actually generate up to 28 fraction plates, which across an 8-column RoboColumn run could essentially create up to 2,500 fractions in it for analysis before we can understand how our separations worked in detail. And this, of course, becomes too challenging for us to analyze using all of our analytics. So we had to come up with a strategic way of deploying our assays by using our tools to their fullest of extent. So typically what we do is that we actually use these on-the-fly measurements and our fast analytics to start overlaying chromatographic traces across the different assays we have deployed so far. And by doing this, we can start understanding where are the interesting areas within our chromatographic separations for each of the columns we have used in our experiment. And this essentially allows us to start defining start and end points of different pools to generate from, again, our chromatographic separations. So what we do next is that we start pooling the different fractions across the different pools that we have defined, and we analyze them using our slower off-tech analytics. And by doing this, we can then understand where are the process reads, information reads, pools within our chromatographic separations. So we go from essentially having to analyze all the fractions to just analyzing a few pools. And by identifying the more interesting pools or fractions, we can then go in them and start analyzing individual fractions with our, again, slow off-tech analytics before we complete our chromatography screen. And by doing this, we have ended up in a situation where we can actually run up to 4 miniature column screens per week when these are being supported by 2 to 3 scientists at a time. So with this, we have actually gone through pretty much all the different workflows we typically deploy within our laboratories in support of our high-throughput chromatography experiments. And we're now going to go through some of the case studies we have prepared for you, showing you some real results we have generated by using all of our workflows. So the first case study is related to actually coming up and developing a capture step for a large envelope virus. So in this case, we're dealing with a rather large virus, was around 250 nanometers in diameter, and it was essentially as complex as it gets. And what happened here is that we had a change in our virus production system, which now was giving us a harvest with a low product titer and rather high impurity levels. And that is why we had to actually focus and start trying to develop a chromatography-based purification process. And the first goal in this effort was to actually come up with a combination of stationary phases and mobile phase conditions for a capture step for our product. So in this case, I would just like to essentially clarify that when we're talking about a good capture step, we don't only mean a step that can actually bind everything. Instead, we should actually try and have a step that can be a bit selective, has elements of selectivity built into it. Because by being selective, we can actually hope to have a higher capacity for our product because there's going to be less competitions for the binding sites from the different impurities. By having these elements of selectivity within our capture step, we can also be able to try and maximize the resolution between our product and the different impurities. And of course, we should not forget that to have a good capture step, which is working bind dilute mode, we should also, of course, make sure that this step can also give us high elution yields, because if it does not, then we will not be able to use it within our process. So what we did during these investigations is that we screened up to 17 different chromatography stationary phases. And this included, for example, different pseudo-affinity ligands, anion exchanges, cation exchanges, and also multimodal resins. And we screened both beaded media and also membranes. And across all of these different stationary phases, we had a good description of different properties on things such as different bead sizes, pore sizes, and also construction material for the different base matrices. And for each of these different stationary phases, we started screening binding conditions and elution conditions, and we also tested some elution conditions with gradients. So we used our high-throughput process development workflows, and in particular the plate chromatography technique, to start screening mobile phases and stationary phases. And for those candidates that gave us the best results, we then evaluated their performance under flow using our RoboColumn chromatography technique. Here we also tested additional stationary phases that we could not test by using only chromatography plates. And we also evaluated the elution yields for our product and also how it could actually be separated from the different impurities under flow. In these experiments, again, we support our own analytical deployment. And here we have a good selection of analytical methods varying from fast to also slow. And for the vast majority of these experiments we carried out during this investigation, our primary product tracking assay was this HT TC50 assay. So this is a relative infectivity assay that is also, again, a cell-based technique, a cell-based assay, and it was deployed in a $1 million integrated robotic system. And with this setup, we could actually analyze up to 500 samples a week versus to only being able to analyze 10 samples a week when this is being run in a manual way. Now, this is mighty impressive, but of course, let's not forget that this is again a cell-based assay, and it was coming in with its own relatively large, high amounts of analytical noise built into it. And this is something that actually complicated our screening efforts quite a lot, I should say. So in this slide, I'm showing you a summary of the different results were generated during our work on this project. So we were able to essentially generate binding maps for both our impurities and our product for each stationary phases as a function of pH and salt. And by doing this, we're able to actually start getting an estimate of binding capacities for our product and trying to figure out if there's any windows of operation in terms of pH and salt, where we could have high binding yields for the product and low binding yields for the impurities, meaning that we bound the product when we're flowing through impurities. For the most interesting combination of stationary phases and again mobile conditions, we also evaluated them under flow using RoboColumns. And here what you can see on the right hand side of these examples from the different chromatographic separations using RoboColumns is that we have overlaid chromatographic traces recorded with different analytical methods. As I said, that we're typically doing within our workflow deploying RoboColumn. So in this case, by doing this, we were able to understand how our product was being separated by the different impurities. And of course, we were able to start estimating elution yields for a product. And also we were able to start understanding rotation trends for a product, which was very useful in terms of trying to design some elution methods based on steps as opposed to linear gradients. And all of these results and other results I'm not sharing with you today were actually generated during a period of 4 months where they were being supported by only 2 staff members. And we were running these in a scenario wherein we had 1 day to perform the screens and then 4 days to analyze our results, to deploy our assays and also analyze results before we could plan the next round of experiments. So it was a rather busy time. So by doing this work across these 4 months, we were able to rank all of these different stationary phases in terms of the binding capacity and elution yields from high to medium and low. And at the same time, we're able to understand how the pH and the salt affect the binding of the impurities of our product. And as you can see from the blue entries in this table, these CX resins cation exchanges actually gave us some rather nice results because they returned both high binding capacities and also high elution yields. And at the same time, because the binding trends for both impurities and for our product were being affected by both the pH and the salt, we could indeed come up with some windows of operation where we could actually maximize the binding of the product and minimize the binding of impurities, meaning that we could actually get the purification just from this binding step alone. However, when we started testing and getting back results based on infectivity, we started seeing that we had a rather large negative effect or drop in our elution yields based on our effectivity assay. And this was because the conditions we had come up with that were making the chromatography work was also having were also having adverse impact, a negative impact on the effectivity of our product. And because in this case we had already started with a production system that was giving us low titers, we couldn't afford to get another hit on effectivity for a product through the purification process. And that's why this time we actually decided not to move forward with this type of chromatography media with cation exchanges. Instead, we chose to focus our efforts on one of our pseudo-affinity ligands or chromatography media and one of our anion exchanges. And the reason behind it was that in both cases, they gave us some high binding capacities and also medium to high elution yields. While we're doing these screening experiments, we also noticed, however, that the binding trends of the impurities for our product as a function of the pH and the salt were actually concurrent, meaning that we were expecting to have a limited potential in terms of selectivity and also in terms of purification, meaning that we could not really find a magic combination of conditions wherein we could again maximize the binding of the product while minimizing the binding of our impurities. Despite this, we chose to actually scale up the pseudo-affinity chromatography media and also the first anion exchanger. And the results we got from this scale-up were actually in agreement to what we have seen from our small-scale experiments. So regarding the anion exchanger, we got an elution yield of around 50% while we had a good reduction of impurities in terms of proteins, but we also noticed we're actually co-eluting hostile DNA with our product. From the pseudo-affinity chromatography media, we got a high elution yields of more than 80% and a rather nice reduction of impurities. However, in both cases, when we tried to estimate their product purity based on STS page, we saw that we're still having rather low overall purity. And what was happening in this situation is that we had started with a large production system that was giving us high titers and a relatively high starting purity, but it wasn't scalable. So we had to go to a new virus production system, and here it was not fully optimized, and that is why we're actually getting low titers and also a rather low purity. Even a 5% in this case would be an overestimate. And as a result, we found out that throughout all of the work we had done in a period of 4 months, that we could not come up with a chromatography step that provide both high yields and also high purity. And this is a negative result, but at the same time was a rather important result, because throughout our work during this period of 4 months, we had generated rather substantial and expansive data packets. And based on this data package, we were able then to make some rather important decisions on a project level. So we're able to essentially justify the allocation of resources in terms of trying to optimize further the new virus production system, and to also try to investigate the prospect of designing some custom ligands for our product. So again, in this case, we didn't manage to find the golden, let's say, conditions to run our capture step, but still all of the work we had done within that period of 4 months was instrumental in terms of making these important project level based, let's say, decisions. The next case study I will just tell you, I will just discuss with you. It's going to be done only in high level terms, and it is quite relevant because it is actually focusing on our efforts to come up with a purification process for one of the 2 COVID candidates that our company is currently working on. So while we're working on this, we were tasked again with coming up with a purification process, and here we deployed our high-throughput process development workflows. And here we had access to up to 7 staff members while using our 2 robotic stations. So we were able to perform up to 7 screens per week that included both plate and also miniature column chromatography experiments. And part of these experiments were also little studies in terms of doing assay transfer, also assay development. So across all of our screens, we were able to screen more than 20 stationary phases, which were also evaluating both flow-through and or binding dilute mode. And during our evaluations, we screened different parameters, including binding elution conditions. We did capacity measurement studies. We investigated the input or resistance time of the performance of our chromatography steps, and we also evaluated them under both a granular step elution operating mode. And we were able to identify the best performing conditions and also stationary phases, and we scaled them up and also verified them. And as a result of that, we were able to deliver 2 1-column processes and 2 2-column processes. And this is all something that was actually done within a period of 2 months. And as a matter of fact, this is a case where we actually started from scratch to come up with a process to actually deliver a purified COVID vaccine candidate in response to the pandemic. And while this was essentially a substantial effort across the entire organization, we were quite instrumental in coming up with different purification process alternatives within only a period of 2 months. So I think this was, in many, let's say, terms, unprecedented within our organization. So to summarize, we have actually spent quite a lot of time coming up with the different tools. We have generated the pieces of software and our automated high-throughput chromatography procedures. And all of these things were actually done so as to be able to essentially create a situation wherein all of these technologies are actually accessible from all of our team members. And by doing this, we have been able to save pretty much entirely the focus our focus on the interpretation of experimental results and the planning of follow-up experimental studies, instead of actually focusing on running the experiments themselves, and even to some extent planning these experiments, meaning in the planning design. So what this has allowed us to do is to have a rather small high-throughput process development team that can support large campaigns of process development experiments and also meet some rather aggressive timelines, like the ones we had to deal with when we were working on the purification of the COVID vaccine candidate. And with this, we have essentially started unhinging the potential of high-throughput process development and essentially trying to have a rather significant impact in terms of improving the process development in the area of vaccines. So before I hand it over to Salome, I would just like to take a brief moment to acknowledge the members of our team, which include both young scientists and some rather seasoned scientists. And without their efforts, we wouldn't be able to be where we are right now. So they have been rather critical and in terms of actually helping this team to take off. So with this, I would like to thank you for your attention, and I will be happy to answer any questions that you may have. Thank you.
Thank you, Spiros, for the great presentation. It was very informative, and for sure it will be very useful to all of us. So the Q&A session is now open, so please raise your hand if you want to ask Spiros a question. Go ahead.
Hi, Spiros. Very nice presentation.
Hey, Marco.
And I have a question regarding your workflows, the one that you have your design of experiments, then you have your results in the end, and then there is a feedback loop almost. So you're performing these experiments in a self-optimization way. How does it work?
That's a good point. No. So this is something we are planning to implement within laboratories, especially in the case of actually doing some upstream process development. But for the time being, this so-called feedback loop is done manually. So every time we actually generate the results from our studies, we need to actually go through them manually before we make the decisions regarding how to continue our experiments. So we don't have an automated feedback loop in this case. But again, this is something we are really having essentially or planning to implement for some of the work we have been doing with upstream process development for cell cultures.
Ellie?
Thank you, Spiros. I haven't heard from you for a long time. Nice to hear from you again. One of the things I wanted to ask was, to what extent do you get a chance to talk to your upstream people? Because looking at the final set of slides you put, especially the conclusion one, towards the conclusion one, it is clear that obviously you can't take something out easily. It's better if we start with impurities being less, or I don't know, titers to be higher, which takes us back one step to the old story of, you can't create something out of nothing. So upstream becomes very important in this respect. To what extent do you get a chance to talk to your upstream people and basically tell them that maybe you're somewhat at the limit of what you can do, and ask them how they can help? Does that happen?
Yes, it does. And as a matter of fact, it is rather important in terms of guaranteeing the success of such work. So we do have open discussions with our upstream colleagues. And typically before we actually start going into these kind of discussions, we need to make sure that we have done everything we can from a downstream processing point of view. So it's not something like, we're going to get some very preliminary results and immediately go back to them and say, hey, we cannot purify these, you need to do something. We make sure we do our due diligence. And if we can't indeed produce something in terms of a downstream purification process, then we go into discussions in terms of how we can actually improve the upstream unit operations and, for example, the production of our viruses. But again, as you mentioned, being able to have this open conversation with our colleagues, it's extremely important in terms of, again, guaranteeing the success of such projects.
Thank you, Spiros.
Adil, please.
Hi, Spiros.
Hello.
Very, very interesting to hear the amount of data you can produce through these high throughput methods. And I was just wondering whether there was sort of more general takeaway kind of information that you could get by, so that you were looking at, you had high binding and you were able to kind of get high recovery of the virus, but then you found it to be low infectivity. So the viability of it was not compatible with the particular method. So the question is, does that translate to other products and do such? So could you build up sort of a body of information that tells you, well, if you're ever going to be working with viruses, membranous, whatever, these type of things, then these are the conditions which are going to be, detrimental to the viability of your product?
So unfortunately, with viruses, you don't have, let's say, one sort of a blanket of conditions or ranges where you're expecting them to have this drop-off in terms of, again, let's say, the infectivity. So you need to evaluate them for each of the different products every time. But the interesting thing about this kind of study was that, and this is something we have started implementing ever since then, part of all of our screens, it's also carrying out small extent stability studies to actually anticipate such issues. So for example, we often implement for each of the different products we're using, some small scale stability studies where we're varying the pH and the salt, and we're trying to find out if there is a negative impact in terms of effectivity. And this has resulted in actually giving us a better idea in terms of how we can actually design our purification processes. And again, depending on the range of the pH, for example, and the information we know about our product, we can also start knocking out even different modalities chromatography stationary phases as well. So it can definitely help us quite a lot when we're doing this kind of process development experiments. But going back to your question, unfortunately, no, there is no, let's say, set range of conditions that we can say that within these range of conditions, our viruses will work or will not work. In fact, in some of the projects, we have been rather surprised by some of our results.
Biology, hey? Fantastic.
Yes.
Thanks a lot.
Yes.
Anyone else? Okay, I have a question for you, Spiros. On your case study 1, you mentioned, well, a large molecule and a very dirty sample. But if you have small molecules and still very dirty sample, are you still able to use your process? Is there any minimal requirements to use your process?
Because these types of technologies, they can be applied to any kind of product, as a matter of fact. It doesn't even have to be from biologics. I mean, people even in the department of small molecules, they routinely use such a type of technology for high throughput screening experiments. The main difference of actually dealing with a smaller molecule will be that things will be relatively easier, even in terms of the type of analytical methods we have and the robustness as well, but even in terms of chromatography, because one of the main challenges of trying to purify virus products or live virus vaccines is that when you're running chromatography steps within a bind dilute mode, we are finding ourselves to be often challenged by elution yields. And this can make things really tricky for us, because even in terms of how we carry out our screens, it means that we don't always go, de facto to a bind dilute step, but we need to evaluate all of our different stationary phases in both flow through and also bind dilute mode. So it makes basically, you're using the same tools and the same processes, but in the case of live virus vaccines, you just need to do more.
Okay. What about the costs of the process? How cost effective it is compared with the normal process?
So this is an interesting question. I mean, if you're talking about, doing some sort of a one factor at a time development of processes, well, of course, because you're running less experiments, it's going to be cheaper. But at the same time, you're generating so much more information by deploying high throughput process development approaches that it actually makes it worthwhile to spend more, essentially, money in terms of running these experiments. And as a matter of fact, even in the first case study that I talked to you about today, even if we did not come up with these magic operating conditions, the fact that we had such an expansive and detailed data package was extremely useful in terms of planning any further, let's say, plan any further activities on a project level. So if we didn't have that in place, it would be difficult to actually justify how we will need to allocate our resources in terms of taking this project forward. And that's something, for example, that if we were to use only this one factor at a time type of experimentation, we wouldn't be able to do at least as easily. So again, we have this very comprehensive data package, and based on that, we could actually justify selection in terms of saying that, hey, we need to work more on improving our virus production system, or we need to invest in terms of coming up with some custom ligands for our product.
Okay, thank you. Anyone else with a question? I guess not. So I will declare the session finished for now. Thank you again, Spiros, for this useful talk, and thank you for giving us this opportunity.
Okay, thank you very much, and goodbye, everyone. Thank you for dropping in.
Bye-bye.
Thanks. Bye.